Good evening, developers of our country. I am Tanisha Paul, reporting from FISC Local News. This just in, induced pluripotent stem cells, otherwise known as iPS cells, are sweeping the country. These cells, as introduced by Yamanaki, are derived from the end cell mass of mammalian blastocysts, having the ability to grow indefinitely while maintaining pluripotency and the ability to differentiate into cells of 3 germ layers. Likewise, since this experimentation, 2 notably newer techniques have been discovered for the generation of iPS stem cells. As adapted from Chiziak et al. Paper 2016, the monolayer culture with defined growth factors and the medium conditioning from the human chondrocyte 402-05A cell line are the 2 fastest and most cost-effective protocols for generating chondrogenic lineage in human-induced primary stem cells from the primary human dermal fibroblast, PHDF. Progress has been made in the area of iPS stem cells only because of the past hurdles that have been cleared. Isis Christopher from 1866 has more on this topic. Isis?
Thank you, Tarisha. Now, since the initial breakthrough of the development of iPS cells, many improvements in this technology has been made. Several of the major hurdles that were surpassed include issues with producing viable chimera, the facilitation of the growth of tumors, and low efficiency. The conversion of iPS cells in the past has been incredibly low. The rate at which somatic cells were reprogrammed into iPS cells in Yamanaka's original mouse study was said to be only 0.01 to 0.1%. With the emergence of innovative techniques, a path has been found to induce efficient reprogramming, near 100% efficiency within 7 days from mouse and human cells. This is done through the downregulation of the nucleosome remodeling and deacetylation complex. Now, the overexpression of MBD3, a subunit of this complex, inhibits induction of the iPS cells. Depletion of this subunit, on the other hand, improves reprogramming, which results in deterministic and synchronized iPS cell programming. Now, depending on the methods used, reprogramming of adult cells to obtain iPS cells may pose significant risks that could limit their use in humans. It was seen that when viruses were used to genomically alter the cells, the expression of cancer-causing genes had the potential to be triggered. However, with the inactivation or deletion of these genes, the efficiency would drastically decrease. Fortunately, in 2011, a new method was discovered, which involved the use of microRNAs, which improved the efficiency of reprogramming without the risk of tumor growth. Now, with innovative techniques to produce iPS cells, efficiency of reprogramming drastically increased, and these cells are now able to be more patient-specific and safer to use therapeutically. Now, a number of disease models has been and is continuing to be tested with iPS therapy. Anastasia Smith has more to this story.
Thank you, Isis. The use of iPS stem cells for disease modeling is based on the fact that these cells are capable of self-renewing. These same very stem cells can differentiate into different types of cells in the human body, which can be utilized for the preparation of different disease models to study the diseases. Genetic disorders, neurodegenerative diseases, and other acquired diseases have been studied using iPS stem cells as disease models. So far, more than 40 iPS stem cells disease models have been successfully generated for patients with genetic diseases, and the length of this list keeps going. A few of these diseases and their features include, but are not limited to, ALS, or Lou Gehrig's disease, is caused by autosomal-dominated mutation in super dismutase and leads to death of motor neurons of the motor cortex, brainstem, and spinal cord. Duchenne-type muscular dystrophy is caused by biochemical and genetic defects in dystrophin glycoprotein complex and leads to loss in walking. Hemophilia A is caused by deficiency in factor VIII and decreases protein production and efficiency in clotting of blood. Back to the studios.
We're going to end tonight's broadcast by expanding on the future advancement of iPS cells. The further expansion of the diseases of which iPS cells are used as a model, for not all diseases have a model solution of iPS cells.
We will also explore new techniques to generate iPS cells with little risk or with the incorporation of vectors.
The continuation of cost-effective and timely ways of generating iPS cells. This brings us to 3D printing using iPS cells. We're going to get further information from Crystal Ruth.
I want to focus on a very interesting area that's combining 3D bioprinting with stem cell regenerative medicine. This is in red because it is only in the early stages, but I think it represents a very exciting avenue through which the field is heading. With improvements in imaging software and technology, we are now able to make accurate 3D images and take scans of body structures inside the body. Using AutoCAD and 3D software, we are able to make CAD designs which can be printed using 3D bioprinters. Now these bioprinters are kind of like the printers you have at home, only instead of using ink, they use special biogels to create the structures that you have in the body. Here, after that, you can seed them with stem cells. Here you see a heart valve being printed, which can then later be seeded possibly with your own stem cells. There's an image on the inset of the heart valve. There's also an image of an ear being seeded with stem cells, which can be your own. Underneath, you see a 3D printed image of a trachea, and on the bottom right, you see an interview that I recently did with CTV National News on the youngest ever transplant recipient of a trachea seeded with her own stem cells. Now, it's important to note that while this is very exciting, it is still in its infancy. We cannot make complicated structures with multiple cell types, and right now it is just very basic. But think about where this can head. Think about whether we could use it in the future to print structures in the body and use our own cells for transplant.
Good night.